The overall goal is to characterize the reducing end and internal region glycosyl sequences of heteroxylans by tagging the original reducing end sugar residue of the water-soluble arabinoxylans and treating with an endoxylanase to generate a mixture of labeled oligosaccharides.
This method can help answer key questions in glycomic field, such as alterations or fine structure of oligosaccharides or polysaccharides in response to plant growth and development, as well as abiotic and biotic stresses. The main advantage of this method is it describes the integrated approach to sequencing of reducing end sequence as well as internal region sequence of any glycan or polysaccharide.
Prepare a 1 molar sodium cyanoborohydride solution in 1 milliliter of water. Then, dissolve 27.2 mg of 2AB reagent in the sodium cyanoborohydride solution by heating at 65 degrees Celsius and adjusting the pH of the reaction mixture to pH 5.5 with 10% acetic acid. Add 200 microliters of the reaction mixture to 1 mg of water-soluble arabinoxylans in a glass tube with a cap and mix using a vortex mixer. Incubate the sample for 2 hours at 65 degrees Celsius in a fume hood. Following incubation, cool the suspension to room temperature and add 4 volumes of absolute ethanol. Place the suspension in a cold storage overnight to precipitate polysaccharides. Then, centrifuge the sample and remove the supernatant. Wash the pellet extensively with absolute ethanol, acetone, and methyl.